Welcome back to the deep dive. Today, our mission is to really get into the nuts and bolts of a core technique in forensic science, DNA electrophoresis. We're drawing from a key chapter in the forensic biology textbook, focusing on how labs separate and measure DNA fragments. If you need that essential understanding of how it all works, well, this is the place.
Absolutely. Think of it as our quest for resolution. Because electrophoresis, it might sound technical, but it's basically the physical sorting step you absolutely need to identify specific DNA fragments. It's crucial.
Okay, let's break that down. So electricity and DNA. Why does the DNA even move? What's the basic principle here?
It really boils down to chemistry, the structure of DNA itself. When it's in water, DNA has a charge.
And that charges.
It's negative, strongly negative, actually. And that's because every single building block, every nucleotide, has a phosphate group, which carries a negative charge.
Right, so basic physics then. Apply an electric field, opposites attract. The negative DNA moves away from the negative end, the cathode.
And head straight for the positive end, the anode.
Now, usually how fast something moves in an electric field, its mobility depends on its shape and its charge relative to its mass, right?
That's generally true, yeah. But DNA is kind of special here, and this is really the key point.
Okay.
Because every nucleotide adds both mass and one negative charge, that charge-to-mass ratio. It ends up being pretty much the same, uniform, no matter how long or short the DNA fragment is.
Ah, okay. So if the electrical pull is effectively equal for all fragments, what actually causes them to separate out?
Size, almost entirely size. We're usually dealing with linear DNA in forensics, double-stranded for the older stuff, single-stranded for newer methods like STRs. So shape differences are minimal.
So it's like a race through an obstacle course.
Precisely. The separation depends purely on how easily the molecule can wiggle through whatever medium we put it in. The smaller ones zip through faster, less getting tangled. The bigger ones, they struggle more friction, so they lag behind.
And that medium is the matrix, the gel.
Exactly. It's the physical support, the molecular sieve, as you called it. It provides that obstacle course and stops everything from just diffusing randomly. The two big ones are agarose and polyacrylamide.
Okay, tell us about agarose first.
Sure. Agarose is a natural polymer, makes a sort of 3D mesh with fairly large pores, think 50 to maybe 200 nanometers across.
Which make it good for?
Big DNA fragments. It's great for separating pieces in a really wide size range, say 50 base pairs all the way up to 20,000.
So historically, that was perfect for things like RFLP analysis, looking at those larger VNTRs.
Spot on. And you can tweak it. Use a lower concentration of agarose, you get bigger pores, which helps separate those really large fragments from each other better.
Okay, so agarose is for the big picture, the larger scale. What about polyacrylamide? You said that's about precision?
Yeah, this is where it gets really impressive. Polyacrylamide has much higher resolving power.
How much higher?
We're talking about separating small fragments, maybe 5 to 500 base pairs. But the resolution is so good, it can actually tell the difference between fragments that differ by just one single nucleotide.
Wow. One base pair difference. That sounds like you need incredible control over the pore size in that matrix. How's that achieved?
It's a more involved chemical setup. You're polymerizing acrylamide monomers and cross-linking them with another chemical, usually called BIS. By carefully controlling the amounts, you can fine-tune that mesh size very precisely.
And that precision is needed for something called denaturing electrophoresis.
Right. Denaturing conditions mean we run the separation using chemicals, like urea or sometimes heat, to make sure the DNA stays as a single strand, nice and linear.
Why is keeping it single-stranded so critical?
Well, if DNA can fold back on itself or pair up in weird ways, its shape changes, right? And that messes up how it moves. If it's always linear, single-stranded, then its movement speed is incredibly sensitive just to its length, its size.
Got it. So that extreme sensitivity is why denaturing polyacrylamide is the go-to for really high-resolution stuff, like DNA sequencing or...
Exactly, and crucially for modern forensics, for separating the small fragments in STR analysis.
Okay, let's shift from the gels themselves to the equipment, the apparatus. We started with slab gels, basically flat plates of this stuff.
Yep, agarose slabs usually run flat, horizontally. You load your samples in little wells near the negative end, the cathode. You add tracking dyes, like bromophenol blue.
So you can see roughly how far things have moved.
Right. The dye runs ahead of the DNA, gives you a visual check on the progress towards the positive end, the anode. And this is typically done under non-denaturing conditions.
And polyacrylamide slabs.
Those are usually thinner and run vertically. And under those denaturing conditions we just talked about, very common for STRs and also DNA sequencing, like mitochondrial DNA. The advantage was throughput. You could run many samples side by side.
But the downside?
They were tricky to prepare, those thin vertical gels. And with samples loaded so close together, there was always a risk of cross-contamination between lanes.
Which leads us to the big innovation, capillary electrophoresis, or CE, getting rid of the slab altogether.
Huge leap forward, yeah. Instead of a flat slab, CE uses these incredibly thin hollow glass tubes, capillaries, often only like 50 or 100 micrometers in diameter.
And what's the advantage of that tiny tube?
Heat, or rather getting rid of heat. The geometry is fantastic for dissipating the heat generated by the electric current.
And if you can get rid of heat efficiently,
you can apply much, much higher voltages. We're talking 200, even 300 volts per centimeter compared to maybe 5 or 10 volts per centimeter on a slab.
Which means?
Speed. Really rapid separation. Runs that took hours on a slab can be done in minutes in a capillary. Plus, instead of a solid gel, the capillary is usually filled with a kind of runny polymer solution that acts as the sieve. Easier to automate and handle.
And detection is automated too, I assume.
Completely. A laser shines through a little window in the capillary, excites fluorescent dyes attached to the DNA fragments, and a detector picks up the signal. That's how you get those characteristic peaks in an STR profile.
Is there a throughput limitation, though, running one capillary at a time?
Initially, yes. Samples were run sequentially, but modern systems use arrays, maybe up to 16 capillaries running in parallel, which really helps boost the throughput again.
Okay, so from slabs to capillaries, what's next in this push for speed and automation? Microfluidics.
Exactly. Microfluidic devices, sometimes called lab on a chip. We're shrinking everything down, controlling tiny, tiny volumes of liquids, nanoliters, in little channels on a chip. Saves reagents, saves precious sample.
And speeds things up even more.
Potentially, yes. Some are modular, like chips that just do the electrophoresis part very quickly, giving you size and quantity info. Think of the Agilent bioanalyzer chip, maybe 30 minutes. But the ultimate goal is integrated devices.
Integrated meaning?
Meaning everything. The whole workflow. DNA extraction, PCR amplification, the CE separation detection, even the data analysis and genotyping call, all on one self-contained cartridge. These are often called rapid DNA instruments.
So what's the big picture implication here?
It means potentially having fully automated systems that can take a sample, like a cheek swab from an RSD, and generate a CODIS-ready DNA profile in, say, under 2 hours. Right there at the booking station, perhaps. It's about massively speeding up the process.
Wow. Okay, but before we get to the super fast future, let's back up. Once you've run the separation, whether it's bands on a gel or peaks from a capillary, how do you actually know the size of the DNA fragment in base pairs? You can see it separated, but how do you measure it?
Ah, good question. You can't just look at it. You absolutely need a ruler. And in electrophoresis, that ruler comes in the form of DNA standards fragments where you already know the exact size in base pairs. You have to run these known standards right alongside your unknown sample.
Okay, so you compare the unknown to the known. What's one way to do that comparison?
A common method, especially with slab gels, is using relative mobility, or TextWar.
How does text work?
It's basically a ratio. You measure how far your unknown band traveled from the starting well, and you divide that by how far a reference point, usually that fast-moving tracking die, traveled.
Okay, so you get a number, a ratio. Then what?
Then you do the same calculation for all your known size standards. You plot the logarithm of their known sizes against their calculated TextWar values. Over a certain range, this plot should give you a reasonably straight line.
And you find where your unknown sample's text falls on that line and read off the corresponding size.
Exactly. You interpolate the size. It's a good estimation method.
But you said estimation. I'm guessing for really precise forensic work, especially with the high resolution of CE and single-stranded DNA, maybe reasonably straight line isn't quite good enough.
You're spot on. That relationship between mobility and size, particularly for single strands, isn't perfectly linear. It's often a bit curved. Sigmoidal is a technical term. So for the highest accuracy, like we need for STR analysis in CE, we use a more sophisticated approach, the local southern method.
Local southern method. What makes that different?
It's about focusing the comparison. Instead of trying to fit one curve across all your standards, it uses an internal size standard.
Internal meaning?
Meaning it's mixed right in with your sample, running in the same capillary lane. These internal standard fragments are labeled with a different color fluorescent dye, so the software can tell them apart from the sample DNA.
Okay, so it corrects for tiny variations within that specific run.
Precisely. And then, to calculate the size of an unknown peak, the method doesn't use all the internal standard peaks. It looks locally. It finds the unknown peak and uses only the 3 closest standard peaks, maybe 2 smaller and 1 larger, or 1 smaller and 2 larger, to generate a very localized, specific calibration curve just for that region. It even calculates 2 slightly different curves and averages them for maximum accuracy.
Wow, okay, so it's really zeroing in on the immediate neighborhood of the unknown fragment using that internal ruler. That sounds incredibly rigorous.
It has to be. That level of precision is what makes forensic STR typing reliable and accurate down to the single base pair level.
Right. So let's just quickly recap this deep dive. We've seen that electrophoresis is the workhorse, separating DNA mainly by size because that charge to mass ratio is constant.
Hmm.
We talked about the 2 key sieves, agarose for the big stuff, RFLP kind of scale, and then high-resolution polyacrylamide, crucial for getting single-base resolution needed for STR.
Right, the scale versus precision trade-off.
And we followed the tech evolution from those manual slab gels, which offered throughput but were cumbersome, to the much faster, high-voltage capillary systems,
Leading towards automation.
which culminates in microfluidics and the idea of these integrated rapid DNA instruments potentially speeding everything up dramatically.
Yeah, and that leads us to maybe a final thought to leave you with. This whole trend we've discussed, from big gels taking hours or days down to capillaries doing it in minutes and now pushing towards these integrated devices. It's really a story about moving the analysis closer and closer to the source of the evidence, taking the lab to the sample almost.
So decentralizing the analysis.
Exactly. And that acceleration, that decentralization,
Mhm.
it raises some really important questions, doesn't it? If you can get a DNA profile almost instantly, maybe outside a traditional accredited lab, how do we ensure quality control? How does that fit with legal standards for handling evidence, chain of custody? What happens when analysis is potentially happening right at a police station?
That's definitely something complex to think about, the downstream consequences, legal and ethical, of this incredible technological speed.
A great point to ponder. Well, thank you for walking us through the mechanics of electrophoresis today. Hopefully you, our listener, feel much more informed about this foundational forensic tool.
My pleasure. Until next time.